This is just an overview of the four human species. So the prodromal syndrome is between the time you get parasites starting to appear in the blood until the time point when you start having those regular fever peaks, and that can be a week or two before you get regular spiking fevers. And it's most often misdiagnosed in the United States as the flu, because you have headache, photophobia, muscle aches and pains, anorexia, some vomiting, sometimes some diarrhea. So it can be misdiagnosed by the physician early in the course of infection. Prepatent period is the time from the mosquito bites you till the time you start to see parasites in the blood. And the incubation period, the time that the mosquito bites you till the time you start having symptoms. And you can see it's fairly consistent among the four species of malaria. So I usually think about 2 weeks from the time that you get bitten by a mosquito to the time you have symptoms. About 2 weeks or so. Sometimes 3 weeks. And fever paroxysms refers to the time point between the different fever peaks. So you can see malaria is different than the other three. Now the duration of paroxysms, so the time you get chills to the time your fever drops back down to normal, you can see is 10 hours, 11 hours, but for falciparum it's 16 to 38 hours, which really takes a toll on the patient. Now, duration of untreated infections. Because of the hypnozoites of Vivax, you can have the relapses for up to 8 years after a single bite of a mosquito. Ovale is not quite as long. It can be approximately 2 years. For malaria, they can have very low levels of parasitemia in your blood, not causing any symptoms, but could cause a problem if you go through immunosuppressive therapy. But up to 50 years it's been found in the blood. And for falciparum, if you don't die from it, then 6 to 17 months of having undetectable parasites in the blood. Now, severity of anemia, it's bad with Vivax, but it's really bad with falciparum. And then involvement of the CNS really is only Plasmodium falciparum. And I'm assuming Dr. Gosnell will talk about this. Nephrotic syndrome it's going to damage the kidneys. This is most commonly associated with malariae, but can occur with falciparum. And hopefully Dr. Gosnell will talk about that too on Friday. Now, diagnosis is thick and thin blood film. So this is a thin film. This is a thick film. It's stained with a Giemsa stain. And then it takes a person looking at it under the microscope to try to find the parasites in the blood. And so in Vivax, Malaria, Ovale, you can see this progression. If you were to take a blood smear every 8 hours, you would see the parasites developing from a ring to a trove to a schizont to a segmenter. And you could see the parasites actually developing over time. That's not true with falciparum. With falciparum, you only really see ring stages in the peripheral blood. And that's because the trophozoites and the schizonts and the segmenters, they've trapped themselves within the capillaries of your visceral organs, the lungs, the heart, deeper portions of the skin, the brain. And so we don't see them in the peripheral blood. We only see the ring forms. So if you were to take blood films from someone with falciparum and you take them every 8 hours, you'd only really see ring stages, and sometimes very early trophozoites, but mostly ring stages. For the gametocytes, they usually take up the majority of the red blood cell, and they'll float around for quite a long time. They don't use up a whole lot of energy. You'll just notice that the falciparum gametocyte looks very different than the other gametocytes. So if you were to see a crescent-shaped gametocyte in a blood film, then you could be pretty confident that you're looking at Plasmodium falciparum. There are commercially available kits for diagnosing malaria. The only one licensed in the United States is called Binax Now. And you put a drop of blood on the filter paper and then add the reagents. And if you see a band form at this level, then it's Plasmodium falciparum. So they've identified an antigen that's unique to Plasmodium falciparum, and you're detecting that antigen on this strip. And then they have what they call a pan malaria antigen also on the strip. And this one will identify, they say, all the other species, but in reality that's not so. And so if this one shows up, then you have a species besides falciparum. Yeah, like this one, this particular test strip would be a mixed infection of falciparum but something else. This pan-malaria antigen can't tell you which species it has. You still have to look at the blood film, but it will tell you falciparum, yes or no, which is what the physician kind of wants to know. Now, having said that, as people start to look at this or start to use this kit more often, they find out it's pretty good at identifying Plasmodium falciparum, but it's not very good at detecting someone that has an infection with 5-Ax ovale or Malaria. So the med tech is still going to need to look at the blood smear, and usually the pathologist will look at it as well. Yeah, so actually these tests are so rapid and they're so cheap and widely available now that even in the field you can make the diagnosis and start treatment without having to go back to the lab, stain the slide, and then find the patient so that you can give them the drug. Now, speaking of drugs, our arsenal of drugs is not that big. We only have one drug that kills the liver stage, and that's primaquine. And we have a number of drugs that will kill the parasite inside the red blood cell. So usually these are active against growing parasites. Quinine, chloroquine are close derivatives. Methlaquine was a collaborative study to develop another drug against malaria. Paramethamine's been around a long time. It's a folic acid inhibitor. But artemisinin, I'll talk a little bit about in another slide, but that was developed by the Chinese that seems to have a good effect on malaria parasites. Now, the interesting thing about primaquine is that it will also kill the gametocyte, so you block transmission. The only thing with primaquine is you got to take it every day. Yeah, so some countries will use it. Sometimes the U.S. and the military, they'll use primaquine in addition to one of these drugs. But most of the time it's given separately. So quinine, its antimalaric property was discovered in the 1600s. A Jesuit priest in Peru learned about this fever tree in which the bark was stripped off the tree and made into a tea that was good for treating shaking chills. And so understanding malaria and chills and malaria, he kind of put the two and two together and thought, well, maybe this would work against malaria. Now, there was no malaria in the Americas, well, at least down in South America in the 1600s. In fact, there was no malaria in all the Americas until it was introduced by the Europeans. So anyway, he sent this bark back to Italy, and they tried it on a group of malaria patients, and they got better. And so that sort of established the link between drinking a tea made from the cicona tree bark and the treatment of malaria. Now, it inhibits hemazoan crystallization, so that means that the parasite can't inactivate the iron, which continues to attract oxygen and eventually damages the parasite membrane. But quinine is very neurotoxic, and so it can affect your hearing in particular. And so quinine is really only used in hospitalized patients, really. Chloroquine, and the interesting story about chloroquine is that the cicona trees were originally from South America, but the Europeans brought it into Southeast Asia and started these big forests of the cicona trees to collect the quinine. But during World War II, the Japanese controlled those islands, and other countries couldn't get at it, including Germany. The Japanese didn't share the quinine with the Germans, so the Germans said, well, we'll just make a synthetic form of quinine. So they developed chloroquine in the mid-1930s. And when the formula was discovered after World War II, then other countries started making chloroquine, and it was cheap to make, and it was pretty effective at the time. It has same mechanism of action as quinine, less toxic, but nevertheless it is toxic. So chloroquine was very widely used from the mid-1940s on. And if you look in Southeast Asia, by the late 1950s, which is only about 10 years, you started to see chloroquine resistance, and it spread throughout the world. So now, if you're going to a malarious area, you need to have consultation on what drugs to take and whether chloroquine works or doesn't work in that area. Chloroquine still kind of works in many areas of South and Central America, but pretty much for the rest of the world, chloroquine doesn't work against falciparum anymore. It still works against 5-X, Malaria, and ovale. And then the Chinese had known about this drug for a long time. It was observed that people that drank this tea called Qinghao Su, which was a tea made for this Artemisia plant, these people didn't get malaria. But it wasn't until the 1980s or 90s that the Chinese put a lot of effort into it and actually isolated the active component, artemisinin, and then developed drugs that could be used to treat uncomplicated malaria. Now, the only problem with artemisinin is that it's quickly eliminated by our body, so you have to take it every day. And also to prevent drug resistance being formed, it's usually combined with other drugs as well. And primaquine, so radical cure means that it cures it from your entire body, which in this case means the liver. It affects the liver form. And it will kill both the schizons and the hypnozoites. So usually in the US anyway, after they've cleared the blood parasites, then they'll come back and they'll give primaquine. But you have to give it every day for 2 weeks in order to kill the parasites in the liver. The only contraindication is people with G6PD deficiencies. So glucose 6-phosphate dehydrogenase is what your red blood cells use to prevent oxidative stress. And if you don't have it, then if you don't have G6PD, I mean, if you don't have, G6PD, then if your red blood cells come under oxidative stress, then they'll lyse and cause intravascular anemia. So apparently permaquine does enough stress, oxidative stress, to cause your red blood cells to lyse. So a person that's going to be receive permaquine needs to be screened for G6PD deficiency. Now DDT really was the hallmark of WHO's effort to control, eradicate, and eliminate malaria from the world. And this graph is a very classic graph because it shows the number of houses sprayed, that is this line, and the number of malaria cases, these bars, to show the inverse relationship. So the less houses you spray with DDT, the more malaria you're going to have. And how this works is you spray DDT on the walls of your home, and after the mosquito bites, then it usually rests on the walls of the house before it leaves to go lay its eggs. And having DDT on the walls would then kill them. So DDT actually is a very good way of controlling malaria. But in 1962, Rachel Carson wrote her book Silent Spring that shows that DDT has an effect on bird eggs in particular. And so many countries started to ban DDT. And that's what's leading to this decline here, is that as countries started to ban DDT, then less houses were sprayed. And so more malaria resulted. And then in this book, published in 2001, Precautionary Principle, they're basically arguing that the benefits of DDT outweighs the risk of DDT to human health. And so what they are saying is, if you live, for example, in sub-Saharan Africa, the risk to your children of dying is quite high. And the risk of you or your children developing a serious adverse effect from DDT is very low. Therefore, in sub-Saharan Africa, it makes sense to start using DDT again. And so people are still kind of debating that now. So controlling malaria or preventing malaria, sleeping under an insecticide-treated bed net is very efficacious. Many philanthropic companies now give out bed nets for free. It's very effective at preventing malaria as long as you sleep under the bed nets, right? Residual insecticide spraying to the houses, so they're trying other insecticides besides DDT. Reducing breeding sites of the mosquitoes, a very difficult thing to do. And then public health education. And this mainly involves, instructing people to seek early help if you think you have an infection. And that's because the longer you wait, the more serious the disease becomes, and you want to prevent death. So if you can get to a health facility early enough and get a quick, accurate diagnosis, and this is where those Binax Now kits come in, a quick, accurate diagnosis, and then given the appropriate drugs can all help to reduce the burden of malaria. So let's talk a little bit about Babesia. And as you can see, it looks a lot like falciparum malaria, and very often it's mistaken for falciparum malaria. The difference being is that Babesia will form these Maltese crosses that will help with the diagnosis. So the natural life cycle of Babesia involves rodents. So the rodents are actually infected with Babesia. And ticks. Ticks are the vector. So instead of mosquitoes, we have ticks, and it's the Ixodes tick that's the vector for Babesia. Now the tick feeds on the mouse and picks up Babesia, and then during the winter months, the tick, well, just before winter, fall, during the fall season, the ticks fall off the rodents, and usually in the United States attached to a large mammal like the deer. The deer do not acquire Babesia, but they keep the tick nice and warm through the winter months, through the snow, so that in the spring then the tick falls off and then can find a blood meal on that small rodent. Now humans become infected if they get bitten by a tick. And within our body, it goes directly, the sporozoite goes directly to the red blood cells. So there's no liver involvement here. Sporozoite goes directly to the red blood cell, and it divides twice. And that's why you can get this tetrad formation. So it'll divide once, and you'll have two, and then it'll divide again, and you'll have four. So if it's in the right alignment, you'll see this cross-like appearance of the infected cell. And then this will break open, and those merozoites will then infect new red blood cells. We can also acquire Babesia through blood transfusions because many people are actually asymptomatic with Babesia, and they might donate blood, and then it gets transferred to another person. And in other parts of the world, again, it involves small rodents, but it might involve other animals, such as cattle. So Babesia divergens is more common in Europe, and Babesia macroti is more common in the United States. But it's basically the same life cycle. Okay, so this is the Babesia. It's going to grow, it's going to divide once, and then it's going to divide twice. And then at this stage, this will burst. So the number of parasites in the blood can be quite low. And so reference laboratories, rather than looking at the blood films, might run a PCR to make the diagnosis of Babesia. What you can do is protect yourself from tick bites. These ticks are the same ticks that transmit Lyme disease. And so you can actually have both Lyme's disease and Babesia at the same time. And drugs are different, so you need a combination of drugs, atovaquone and erythromycin, to get rid of Babesia. And so prevention is avoiding tick bites by using repellents such as DEET, avoided woody, brushy kind of areas with high grass where the ticks can move from the environment onto your skin. And that's why it's recommended that if you're going to walk in kind of wooded areas, that you walk along trails to avoid picking up ticks, and then finding and removing the ticks as soon as possible. So here's a case study. 24-year-old Cameroonian woman. Cameroon is on the west central side of Africa. Came in for nausea and vomiting. The clinic determined she's pregnant for the first time. So there's going to be a concern about the effect of malaria, especially falciparum malaria, on her pregnancy. And blood films for malaria was ordered, and the following was found. So, okay, so you can eliminate Plasmodium knowlesi because it's found in Southeast Asia. You can eliminate Vivax potentially because it's found, it's not found in Africa, or very rare. Oops. And so the answer is Plasmodium ovale. It's not falciparum because it's older than a ring. So this 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 parasite is quite large trophozoite, maybe early kind of skyzont. Plasmodium malariae, it likes to invade older red blood cells, so you might expect a small cell like this to be infected, but not a larger cell. And then finally, it has these Schuffner stipplings and kind of an oval shape. They give it away as ovale. So at this point, they wouldn't be too concerned with her her pregnancy, and then she could be treated appropriately. I'm going backwards.